Now it's time for the session with polar research highlights from other universities. And we will start with Professor Anna Wåhlin. Anna, you're there. Welcome up here on stage. Anna, you are Professor of Physical Oceanography at University of Gothenburg, and your speech will be about observations from below an Antarctic ice shelf in 2022. That is very recent data.
Thank you. I'm going to show some results that we got from the cruise in Antarctic, last Antarctic field season. So this was in January, February, and March 2022. And we went to Thwaites. The goal was Thwaites Glacier in West Antarctica, the Amundsen Sea, and we didn't quite make it. Instead, we had to go to the sister glacier, Dodson and Getz. And on board, this was from the US icebreaker Palmer, and we had our AUV Ran, the orange thing here, and there was also the UK AUV Boatimac Boatface, the yellow thing. And yeah, I'm from University of Gothenburg, and the other people on board are listed here from Sweden and UK and US. This expedition was part of the international Thwaites Glacier collaboration, which is the largest field program that has been funded to Antarctica so far. It's in total 25 million over 5 years, and the main field seasons started in 2019, and we are just coming up now on the last field season, which is mainly on land. So it's a combination of ocean work, submarines or autonomous underwater vehicles, and also lots of things done on land with radars and drilling and stuff. In this program, there were funded 7 different projects, and I'm a member of Tarzan project. So this is what we did. This is the ship track. It's hard to... Can I point somehow here, or not really? No. So we, after giving up, Thwaites Glacier is further east, and we tried to get in, and quickly we didn't even try. We looked at the ice maps and realized this is not possible. So then we came in here, and the target was Dodson Glacier, which is down here. And we did lots and lots of CTD stations. All the yellow dots are CTD stations. In total, 269 casts. And on the CTD, we also had LADCP, so that is a current meter that you mount on the CTD that was triggered with it. And you can see here the results. So there's an inflow here in the west. That one is deeper down. And then there's an outflow in the east of meltwater-rich water formed under the ice shelf. In addition to CTD, we also had 7 gliders on board. And here you can see the tracks of those gliders. They measured also temperature, currents, salinity, and some of them had some quite fancy biogeochemistry sensors on board. And this is the results from AutoSub long range, also known as Boatimac Boatface. So on this, this is a collection of all the missions that was done. Boatimac Boatface was supposed to go quite far into the cavity. So the black line up there is the ice edge. And they went in one straight line and then back out. And then they did that, repeated that 3 times to make this sort of trident. So here is temperature, about 100 meters from the seabed. And this is velocity, zonal velocity and meridional velocity. So the main takeaway here was that indeed this ice shelf cavity has quite warm water on the seabed, and the velocities are varying quite a lot. You can't see a distinct, strong inflow or outflow near the seabed. It seems to be more complicated than that. And we also had this, this is the RAN, the AUV that we have at University of Gothenburg, and this is a infrastructure that is open, so anyone can use this if they want to. And the good thing with RAN is that it's very good navigation. Navigation is a big challenge for these underwater vehicles, and this is from tests that we did in Härnösand a couple of years back, where we tried the navigation under ice. So it's more difficult under ice because you need to have an upward-looking acoustic sensor that measures speed over ice. And here we wanted to test if it worked, and we were very happy with the results. So here it came up. We didn't have a ship. We launched it and recovered from the dock in Härnösand, and this was after 8 hours under the ice. So we were very happy with that. So the main things, Plan A, what we wanted to study was a continuation of what we learned in 2019, the first field season on this project. So using RAN, we sent RAN in, in this 2 of the deep troughs that lead into Thwaites Glacier, T2 and T3. Very fantastic names. So since we could send it under, we could detect water coming from the east, and then we figured out that yes, indeed, there is a through flow in this glacier coming from the east, where you have Pine Island Bay and so on. So that showed previously unknown route for warm water. And we also learned that the warm water coming in from the east is lighter than the warm water coming in from the north. And since this is situated deeper down than the one coming from the north, we also understood that somewhere here there is quite vigorous mixing going on when they meet each other. So Plan A was to look closer at this and send the sub in again from the east and repeat the sections we did from the north. And we also wanted to have a closer look at the bathymetry, because if you have high resolution data from the bathymetry, you can figure out what happened the last time this part was iced over. And in 2019, we also managed to get some of that high resolution bathymetry. So here we could see how the ice has been scraping over the seabed when it was grounded here, and also the small scale features. You have the scraping, the ice has moved upward in the image, and in addition you see these ribbed features, and they are likely caused by tides. So using that information you can calculate how quickly the ice receded from this area, and it turns out it was nearly 3 times faster than it is presently receding. And these tracks were made about 100 years ago. So that was also new information that will be used to guide models of this region in the future. So that was Plan A2, to come back and cover more of these areas with high resolution bathymetry. And here's a picture of the AUV being lifted on board Palmer. It lives in a 40-foot container, so it's a big operation. It's not so common with 40-foot containers. It's quite heavy, and it takes a lot of space on the aft deck, and it's difficult to transport also. So it's hard. Not every harbor can handle a 40-foot container. And inside the container is the AUV and also a launch and recovery system, which is like an arm, the cradle that goes out like this at the aft of the ship, and then it tilts, so the AUV slides down into the water. And when you recover it, you do the same operation in reverse. So you winch it up through the cradle, lift it up, and then go back inside. So it's very neat. We were very, very happy with the way it worked. It's much safer compared to the previous launch and recovery system that we had. Here's a picture from the sea, what it looks like when you during launch. So, using this launch and recovery system, and giving up on Thwaites, we managed to go to Dotson ice shelf, where we did in total 14 missions, and in total 1,075 kilometers under the ice. And this map here shows all our missions under Dotson. And you see these, how we have built like lawnmower patterns, and that is to make a good map of the underside of the ice with our upward-looking multibeam that we have mounted. And here's an example of what it looks like. So this kind of map we have not seen before. We were very astounded when we saw it on the ship. We weren't sure if it was going to work or not. And then the next fear was that maybe it's all flat. Maybe we won't see anything. So instead we saw these features, and it's different features in different sides of the ice shelf. And we don't know. We are working together with glaciologists to try to understand what it is we are seeing. You see the cracks coming across. They are also visible on the surface from satellites, so they go all the way through the glacier. And these swirly patterns here are either caused by ocean water coming in and melting the ice, or they are remnants from thousands of years ago when this was grounded, subglacial hydrology features. In addition, because we did these lawnmower patterns, we also got patterns of other things in the water column, and this one shows fluorescence, which can be converted to chlorophyll. So the high fluorescence means there is productivity in the water. And this picture shows dissolved oxygen. So high productivity goes with high dissolved oxygen, which is primary production. But this is 10 kilometers in. There's no sunlight here, so we were surprised to see that. This water must have come from the outside or being formed in one of those cracks that we saw. We also could see very high productivity. This is from the front of the ice, and it's clear that the water here is super productive, even compared to a Polynya standard. You see the brownish tints here, so algae are growing on the ice itself. And we also, another trial that we did was to collect water samples under the ice, and we had a team of chemists on board, and they were very happy to get water samples from under the ice. So it was a bit of a trial, and one thing is that we always ran out of water. It was so popular. Here you see how they are first low ice sampling for rare metals, I think it was. No, sorry, it's vad heter ädelgas? Neon, noble gases, yeah. And then it goes on to other things. So they took the leftover of Laura's water, and then it went on to the next and to the next. So they were like treating it like gold, this water. Yeah, oops, sorry. No, we were done. That's it. Yeah. Thank you.
Thank you, Anna, for this fantastic presentation. So please stay here.
Yeah, okay.
I guess there are some questions. I have a question. Did I get it right that the tracks that looked like motorcycle tracks were about just 100 years ago?
Yeah, that's right.
100.
Yeah.
And then you said that the new patterns of the chlorophyll and the tracer oxygen, was that in the same area? And does this mean that this ecosystem species, this species are quite new in this area?
No, sorry. No. For being unclear, it was different. The motorcycle tracks were from 2019 outside Thwaites.
Yes.
And the chlorophyll was from Dotson ice shelf from this year.
Yes.
Yeah. Near the ice. The motorcycle tracks are on the seabed, but all this was near the ice.
Yeah. Okay. Questions from the audience? Yeah, Margareta.
Just ask, so what, you obviously went with the ship. It did not look like Odin. No?
No. It's US icebreaker Palmer, NB Palmer.
Yeah. And how do you access that as a Swede?
We got invited to come along, so they are supporting Swedish
world-leading marine research. Yeah.
Great, thanks.
More questions? Okay, Anna, I guess you already have one of those, but here is one extra
actually.
for you.
Thank you very much.
Okay, you earned it. Thanks for coming. Now we invite Professor Love Dalén. You are professor in evolutionary genomics at Stockholm University, Love. Welcome. And your speech will be about evolution of the woolly mammoth.
Yes. Can you hear me? So first of all, thank you for inviting me to give a talk here. I'm glad I could make it finally. It didn't look like I could come because family reasons, but it opened up in the last week. So I'm going to be talking to you about evolution of the woolly mammoth, and more specifically, I'm going to be talking about going from the origin of woolly mammoths until their extinction, which is kind of a journey that spans roughly 1 million years. And I work at the Center for Paleogenetics in Stockholm, and this is a relatively new center. We moved in, or we opened the center in 2020, in February, right before COVID started. And when we moved in, we were just 16 people, but thanks to a lot of very nice funding from VR and the ERC and the Wallenberg Foundations, we're growing very fast. We have a lot of external funding, and we are now 5 research groups, and sometime next year we will hit 45 people in the center. So that's fantastic, and it's a lot of fun to see so many different research groups with different kind of themes joining up in this center, everything from geologists to archaeologists and zoologists like myself. All right, so if we're going to talk about the origins of woolly mammoths or late Pleistocene mammoths, we need to first have a quick look at the sort of classical model of mammoth evolution. And so the way it goes is that if we rewind the tape about 2.5 to 3 million years ago, we have the southern mammoth, Mammuthus meridionalis, which is considered to have been a fairly temperate species of mammoth. At some point in time, this model says that these southern mammoths evolve into steppe mammoths, Mammuthus trogontheri. And this was a kind of dominant species during the early Pleistocene. It inhabited the Arctic. But then the model says that at some point in time, about 1.5 to 800,000 years ago, the steppe mammoths diverged into 2 species which are more famous among lay people, which is on the one hand the woolly mammoth that inhabited in the northern hemisphere in the high Arctic, and then the Colombian mammoth that inhabited the lower 48 states of the United States, as well as down into Mexico and Costa Rica. And so we have for a long time wanted to try to disentangle this speciation event. And so the idea has been, that we've been carrying with us for many years, has been that we would be really nice to go back in time and get DNA from before this speciation event and right after it, because then we can test ideas about the rate of evolution, and we can try to investigate when this separation happened, etc. And so back in 2021, we finally published a paper where we had done that. we had 3 samples. These are woolly mammoth, or these are mammoth teeth. The one at the top is from one of the earliest known woolly mammoths, the Chukotka mammoth. It's about 700,000 years old, give or take. And then we had 2 steppe mammoth teeth that were somewhere on the line of 1.1 to 1.2 million years old. And we did manage to get DNA from these. The DNA in samples of this age, beyond 1 million years, is extremely degraded into very small pieces. But nonetheless, as you can see here in this figure, the gray part in the distributions are DNA sequences that were long enough so that we could trust them. And the red parts in the distribution are DNA sequences that likely, to a vast majority, are mammoth, but we consider them too short to trust. We just can't be sure that they are mammoth and not bacteria or human contamination, etc. Okay, so to make a fairly long story short about the origin of mammoths, on the left-hand side here you can see a time axis showing the sort of middle Pleistocene and the parts of the early Pleistocene. And so our samples are shown, the color is a bit off here, but they are shown up here in green here. This is the phylogenetic tree of mammoths and Asian elephant. And if we place in our 3 samples on the tree, there are 2 things here that are important. One is that on the right-hand side, the Krestovka mammoth, which we thought was just an ordinary steppe mammoth, turns out to have been something completely different. So if you look at the steppe mammoth and the Krestovka mammoth, these inhabited northern Siberia at roughly the same time, about 1.1 to 1.2 million years ago, but they clearly belong to completely different lineages. Probably different species. I'm not allowed to say that, but I'm going to say that anyway. And it turns out, when we do population genomic analysis on this data, that the Colombian mammoth is actually a hybrid species, where about 50% of the genome in the Colombian mammoth is from the woolly mammoth lineage, and 50% comes from this previously unknown taxon of mammoth, the Krestovka mammoth. So that was a pretty big finding. Then the second thing in this paper was that since we had a steppe mammoth that preceded the origin of woolly mammoths, and we had an early woolly mammoth, and then we have also genomes from woolly mammoths from the late Pleistocene, we have this kind of temporal transect through time that spans the origin and the evolution of the woolly mammoth. And so you can see that here you have the Adycha steppe mammoth at 1.1, you have the early woolly mammoth at 700,000 years, and then we have, I think it was 5 mammoth genomes that were between 50,000 years old and 4,000 years old. And so this allowed us to ask the question, during the speciation event of the woolly mammoth, did adaptive evolution happen faster or not? I thought it would, but I was wrong, as I usually am. So it turns out when we look at non-synonymous mutations, these are mutations that lead to a change in the protein sequence, as well as when we look at mutations, non-synonymous mutations in genes that are, in previous papers, have been identified as underlying genetic adaptations to the Arctic environment in mammoths. We can basically draw a straight line through this, so there's no increase in the rate of adaptive evolution as far as we can find across this speciation event. That's interesting in itself, and we are now working on looking at different aspects of this. All right, so that was the origin of the woolly mammoth and as well the Colombian mammoth. What about the extinction? Well, the woolly mammoth had a very wide distribution during the late Pleistocene, shown here in dark gray. We know that they disappeared from this distribution, or at least most of it, at the Pleistocene-Holocene transition. But there was one population that survived quite far. There are reports of other mammoth populations that might have survived fairly far, but as far as we know, the longest surviving population of woolly mammoths inhabited Wrangel Island. Now, Wrangel Island is a fairly small island if you're a mammoth, not if you're a human, and it's also not very productive because it's very far north. So when we use ecological equations to compute how many mammoths there could have been at Wrangel Island at any given point in time, the answer is probably less than 1,000 mammoths. So this is a very small population that survived for 6,000 years before finally going extinct. And so we have been working trying to investigate what happens with the genomes of these mammoths through time. And thanks to support from Polar, we were able to also go to Wrangel Island in 2017. This is a photo from that expedition where we're trying to find mammoths. It was a broader expedition with many different aims, but my component in this, together with a couple of colleagues, was to specifically find mammoth fossils. We also collected sediment samples in order to try to see if we could get mammoth DNA from these sediments. We enjoyed the local wildlife, a bit like a safari. But we did also find mammoth remains, such as this one from the Gusinaya River, and this made us very happy. This is Patricia Pechnerova, who did a PhD with me on woolly mammoth genomics. And so, the question we want to ask is, did genome erosion, so genetic problems, contribute to or even cause the extinction of the woolly mammoths on Wrangel Island? The theory of this is that when you have a large population that becomes small, you will have an increase in genetic drift, leading to loss of genetic diversity. You will have increased inbreeding, and this can lead to inbreeding depression. And also, in theory at least, you can also get an accumulation of deleterious mutations. So these are mutations that cause genetic diseases. And by then sampling late Pleistocene mammoths from the large population and comparing these to these Wrangel genomes, we can then test these hypotheses. And so if we started looking at genetic diversity through time, this is a picture, it's the late Pleistocene, the wiggly thing is the temperature curve or the proxy for temperature. These dots are individual mammoth genomes. And as you can see here, during the late Pleistocene, the last ice age, genetic diversity was relatively high and it was stable. But when we move into the Holocene and Wrangel Island, there is a massive drop in diversity. And if we zoom in on Wrangel Island, we also see that during the Holocene, there is a kind of gradual loss of genetic diversity. It's not large, but it's there. So this is caused by genetic drift. Second question then about inbreeding. Well, neat thing with genomics is that we can measure inbreeding levels in individuals. And the way that works is that if we have this, is just showing a part of a chromosome, each number here is 1 million base pairs of DNA sequence. And if we look at this outbred genome, there is diversity across the chromosomes. But if we then compare that to an inbred genome, we find these long chunks, many millions of base pairs where there is no variation between the two sister chromosomes. That's because of inbreeding. So we can compute the inbreeding level at an individual basis by just estimating the percentage or proportion of the genome that is comprised in these runs of homozygosity. So we do this, we have 23 mammoth genomes, we plot that through time, and what we find is that during the last ice age, we have very little inbreeding, and then it goes very high, up to 40%. This is higher than the Swedish wolf population. And then nothing really happens. It increases a little bit. But the big change happens at the Pleistocene-Holocene transition. Then, final question, what about these genetic diseases? These are preliminary results. None of this is published. When we look at mutations that are classified as being highly damaging, what we find, in contrast to previous papers, is actually a decline in these highly deleterious mutations through time. So moving from right to left, it's not crystal clear, but there is clearly, nonetheless, a decline in these highly deleterious mutations. This is likely caused by inbreeding depression. So natural selection is getting rid of these highly damaging mutations. That is also not good for the population. It's not entirely happy. At the same time, if we look at only slightly deleterious mutations, they are instead increasing. And this is exactly what we would expect from theory. And it's interesting. But having said that, if we summarize all this, the picture that we're seeing is that yes, mammoths were affected by genome erosion during the Holocene, but if genome erosion had caused extinction, I would have expected that during the last, say, 500 to 1,000 years, things would have started the population should have getting smaller and smaller and smaller, inbreeding should have gone even higher, etc. That is not the picture we're seeing. So we don't think that inbreeding problems caused extinction. We can't be entirely sure, but it looks more that we have a fairly stable population that suddenly goes extinct at 4,000 years ago, and we don't know why. Okay, So finally, I just wanted to quickly mention that we have been, in the past, in my research group, focusing a lot on the extinction. What we are now going to do in the future is to focus on a new research program called Deep Time Genomics, where we are gotten funding from VR and the ERC, and we're going to be looking at these kind of deep time types of questions. So we want to investigate the tempo and mode of speciation. We want to look at the importance of past hybridization events. We want to look at the adaptive evolution through time across speciation events, as I mentioned. And we also want to look at the impact of past environmental change. And we're going to do this not only on mammoths. We're not done with the mammoths yet. There's more to do. But we're going to expand this across a number of different taxa from the Arctic, including collared lemmings, voles, horses, bison, muskox, moose, and rhinoceroses, and probably a number of more species. And with that said, I thank you for listening, thank the funders and all the people involved in this work, and I'm done. Thank you.
Thank you very much, Love. Impressing, I must say, really. Questions from the audience here? Magnus?
Two questions, Love. When you looked for mammoths, I'm sure you found remnants of other animals too. Did their populations change in Wrangel Island?
Funnily enough, no, there are only two properly terrestrial mammal taxa on Wrangel Island. It's the lemmings and mammoths. And for all we know, these are the only ones that survived into the Holocene there. Of course, you have Arctic foxes and polar bears. There are finds of woolly rhinoceros, there are finds of horses and bison, but every time these have been radiocarbon dated, they are older than 10,000 years.
How about human colonization of Wrangel Island? Human colonization, any remnants?
Yeah, so there's only one archaeological site on Wrangel Island. It has been tentatively dated to about 3,500 or 600 years ago. So we think the humans that arrived there actually came there after, a couple of centuries after the mammoths went extinct. Also, this is a summer camp where humans came, killed a bunch of geese, a couple of walruses, and then left. We searched heavily for other signs of humans having been there and hunting the mammoths, but to no avail.
Okay, thank you.
Hi, super interesting. Wasn't part of your story that the mammoths on the island was unrelated to the mainland mammoth?
No, they're of course descendants of the mainland population. We're not entirely sure exactly from where, because there's a gap in the fossil record in mammoths in northeastern Siberia, so it appears that they disappeared 12,000 years ago, and then they suddenly show up on Wrangel Island 10,000 years ago. Yeah. We think that the Wrangel Island mammoths probably originate from a population that was even further north in Beringia, which is now underwater. Perhaps New Siberian Islands, north of the New Siberian Islands, etc. But it's difficult to test unless we go and dive and dig up bones from over there.
But here you were dependent on working together, collaborating with people in Russia?
Yes.
Again, so you are also in trouble now with war?
Yes. I mean, obviously we are still collaborating with individual scientists in Russia with the material that we have and the work we're doing, but we have halted all plans for field work and sort of official collaborations with institutions, etc. And we are, for the future projects right now, all our focus is in North America, so Alaska, Canada, and to some extent Greenland.
More questions? No? Okay, Love, this is a hat for you, and thank you for coming here.
Thank you very much.
I think you need it, because congratulations to the fantastic founding that you have received and the worldwide.
I couldn't afford to buy a hat.
No, I mean that you will be in field very much.
I will.
Maybe you will use the money for other things. Congratulations to the worldwide recognition of your work. And I'm so curious of the next woolly mammoth chapters.
we will see.
To be continued.
We have we made a list of all the ongoing projects, and I think we have like 15 ongoing mammoth projects right now, so there will be more.
Okay.
Yeah, thank you.
Thank you, Love. Next speaker is Hanna Farnelid, Senior Lecturer in Marine Ecology at Linnaeus University. And the title of your work, Hanna, presentation is Primary Producers in a Changing Central Arctic Ocean. Welcome.
Thank you. Thank you very much for the invitation. It's just very intriguing to be a microbial ecologist and give a talk right after a mammoth talk. So, I will talk to you today about work that we conducted on board the Odin last summer during the Synoptic Arctic Survey expedition. And I will take you into the microbial world and talk to you about phytoplankton, which are primary producers in the ocean. So we will start under the microscope. So these are microscopic images from our lab in Kalmar with Arctic strains that we isolated during the expedition last year. And these primary producers in the global ocean are responsible for half of the oxygen that we breathe. So they're very, very important in the ecosystem. And it's quite amazing that these tiny, tiny organisms are so important to our everyday lives. And this is on a micro scale. And as you see, most of these phytoplankton are very, very small. And I will return to that later during the presentation. So the Central Arctic ecosystem is the largest Arctic marine ecosystem. And it is covering an area of 3.3 million square kilometers, which is an area more than 7 times the size of Sweden. And it has permanent ice cover both in winter and summer, and that's why we need powerful icebreakers in order to do research in these areas. But the Central Arctic Ocean is also an ecosystem that is undergoing a lot of change. So with global warming, the permanent ice coverage will be transformed into more seasonal ice coverage with the ice edge moving further north during summer. And the melting of the ice will create novel habitats for organisms that live in these areas. There will be more fresh and brackish water habitats, which could have large consequences for the species that sustain. The warming will also cause changes in stratification and nutrient availability, which will have large impact on phytoplankton growth. And in these changes, there will be changes in the overall biological production, and we can also expect changes in the distribution and the composition of species that inhabit these areas. So, in the Central Arctic Ocean, we have some idea about the physics and the chemistry of the system. But an ecosystem is so much more. It's an interplay of interactions between chemistry, physics, and biology. And knowledge about the biological part is still largely lacking. And phytoplankton are really in the center of the food web, forming the base of the microbial food web. And therefore, knowledge about phytoplankton is essential in order to predict what ecosystem consequences will be by global warming. So just to give you a perspective on the biological measurements that have been done in these areas, to your left there is a map showing chlorophyll samplings in the Arctic area. And on the right, there is a map of zooplankton samplings. And I have put yellow circles in the area that we visited during last summer. And as you can see on this map, this is really an unexplored area when it comes to biology. So the Synoptic Arctic Survey was an international collaboration with many different countries participating. And because Sweden had the infrastructure with icebreaker Odin, we had the task of visiting these most difficult areas to do research on. So here's also a map of the cruise track. And as you can see, this is very much corresponding to the yellow circle that I had drew in the previous slide. So we are really in novel territory and taking measurements for the first time, exploring primary production in this area. Going to an unknown area, we were asking very, very basic research questions when it came to the primary producers. And these pictures are members of my team on board. Christian Leiber, Lisa is here today, Hans-Jürgen is also here. And we had a really big team working with biology and primary production on board, so we could really cover large different research questions. And I think this was the effort that was made during the expeditions was really unique in this sense, in that we had a very large biology work package on board. So very basic questions. How many were there? How active were there? Who is there? So sampling DNA and RNA, looking at what are they doing, what processes are they participating in. We also performed experiments looking at grazing rates, nutrient uptake rates. So how do they die and what do they need to grow? And as you saw with the cultures, we're also working on physiological characteristics of cryophilic strains. And with all this data, we hope to combine it in order to get a holistic view of the ecosystem production. So thinking about ice and ice habitats is quite intriguing. And these are some of the species of phytoplankton that we were expecting to see. So diatoms and green flagellates, dinoflagellates, and picoplankton. And in the ice, as the ice is melting, there are brine channels producing brackish water. And these are very, very important habitats for phytoplankton, because then they become a food source for larger organisms. Here's a wonderful picture of Christian in the middle doing some ice core drilling to look at phytoplankton in the ice cores. And to look at the chlorophyll content in different parts of the ice core, we divided the ice cores into different sections. And chlorophyll is then a proxy for the total phytoplankton biomass that we see in the ice core. Dividing the ice core into different layers, ranging from the top snow layer to the surface layer, and then down to the bottom. In this figure, you can see to the right that the chlorophyll content is the highest at the bottom of the ice. So underneath the ice, on the surface of the sea ice, is a very important habitat for phytoplankton. In addition to the sea ice measurements, we also did water column measurements. So we had a CTD instrument both at the aft and the bow of the ship. So with this two systems, we were able to get a very high resolution coverage also of the water column. And in this figure, you can see the depth on the y-axis and the cruise track distance on the x-axis. And these are measurements of total chlorophyll along the cruise track that we did. And we observed a chlorophyll max, commonly around 20 meters depth. The concentrations of chlorophyll were consistently below 1 microgram per liter. And just as a comparison, in the productive season in the Baltic Sea, typically we measure between 1 and 5 micrograms per liter in chlorophyll. So in total, there was very low chlorophyll A in the pelagic Central Arctic Ocean, as we had expected. When it comes to phytoplankton, they can come in many different shapes, forms, and sizes. And we like to divide it into different size classes. The larger phytoplankton we can sample, for example, using a multinet like you see in this picture. So in this multinet we can capture phytoplankton at different depths and study what inhabitants are at different depths. But most of the phytoplankton are in the smallest size fraction. Those are called nanoplankton and picoplankton. Those are often defined as phytoplankton that are below 2 micrometers in diameter. So if you remember your school rulers and you look at the millimeter scale, divide that into 1,000, and that's about the size of a picophytoplankton. And when we did our chlorophyll measurements, we studied what contribution was from the total phytoplankton and what contribution was from the picophytoplankton. And these are our results summarizing the total observations during the expedition. And here in yellow showing the larger than 2 micron contribution, and in pink the 0.3 to 2 micrometer contribution. So the largest contribution to the total chlorophyll was from the picophytoplankton. And if we look at the same chlorophyll figure that I showed you before, but then look at the contribution from the picophytoplankton on the bottom here, the red color is indicating a contribution of more than 80% to the total chlorophyll. So the majority of the phytoplankton in the Central Arctic Ocean were the smallest phytoplankton. And increasing the resolution using flow cytometry techniques, we could measure picoeukaryotes, which are small eukaryotic photosynthesizers. And we found abundances of 2 to 20,000 cells. And frequently, these picoeukaryotes, they composed more than 80% of the total phytoplankton cell counts. One of the hypotheses that we had when we went to sample in the Central Arctic Ocean was that there was a process of Atlantification, with novel species entering the Central Arctic Ocean from the Atlantic Ocean. And one of the species that we were looking for was the Cyanobacterium synechococcus. But in our data, we find no evidence of intrusion of synechococcus into the Arctic Ocean. But model predicts that in a future scenario, there will be more picophytoplankton in the ocean, or in the Arctic Ocean. So in this map, you see the climate change predicted impact on picophytoplankton biomass. And the region of the Arctic is the region on the globe where picophytoplankton biomass is expected to increase the most. So to summarize, studying primary producers in a changing ecosystems, we found a very low primary production rates. We can expect more and higher proportion of picophytoplankton in the future. And with the ice melting, we can also expect new habitats and possibility of new species of phytoplankton. And the consequences that this will have for the ecosystem is that a shift to smaller phytoplankton would mean less nutrition and more energy losses in the food web. So a shift to smaller phytoplankton can have large consequences for the ecosystem and the food web. So with that, I would like to say big thank you to funders and everyone that was participating on board the expedition. And sunset in the Arctic.
Thanks a lot, Hanna. So impressive presentation about your results from the SAS expedition. Thank you very much.
There will be much more results.
Yes, we are looking forward to that too. Are there any questions from the audience on Hanna's work? Okay, I have one question. It was a bit quick in the end, so perhaps I didn't get all of your points. But when I heard you say that there will be an increase in zooplanktons due to climate change, I thought that, okay, there may be an increase in species up the trophic chain, perhaps more fish. But your conclusions was the opposite. Did I get it right?
So if there is a shift towards smaller phytoplankton,
yes,
then there will be more levels in the food web. So there will be more losses also in the food web. So smaller phytoplankton could mean less food for larger species like fish.
I see. Thank you for explaining this. Thank you. Okay, just a minute or a second. Welcome back. I hope we will welcome you back on Odin again. Thank you very much. So next presenter is Associate Professor Ellen Dorrepaal, and you are Associate Professor in Arctic Ecology at Umeå University. And the title of your speech is Year-Round Climate Feedbacks of Winter Phenological Mismatches of Plants and Microbes Across the Arctic. Please, Ellen.
Thank you, Katarina. Yes, I changed the title on my slide a little bit because I realized that was a bit of a mouthful, and given that we were asked to give a presentation that was understandable also for people outside of our field, I decided to change it into maybe another still hard to understand title, but maybe just focus on the last part, redefining seasonality, because that's what we're working on at the moment. So yes, I am officially working at Umeå University, but since 2010 I'm full-time based in Abisko, and I have the privilege to basically be year-round at the Abisko Scientific Research Station, which I enjoy very much. But ever since then, you know, everybody who I meet is always asking me, oh, you're living up there, how is it in the winter? Isn't it long and cold and dark? And yes, I mean, there is a time of the year when we don't get much sunlight. But actually, I got really fascinated by this, and I want to tell you a little bit about the background of why we started to look at redefining seasonality. And that is some research that we did before on permafrost ecosystems and thawing permafrost ecosystems. And these are some results from rooting depths at different depths, actually in the snow fence experiment that Margareta is doing at Storflaket. And here I only show, oh, there is no pointer here. So I have some colored rings there. So you can see this is results from the deep thaw treatment. So we are thawing the permafrost. So the upper two panels show the rooting depths through the season, through the summer and then winter season and next spring in the 0 to 30 centimeters and the 30 to 60 centimeters. So that is what is the active layer in the normal plots. And then the lowest panel actually shows roots that are growing below 60 centimeters. And what you see here in this red circle is that there are really in the end of the growing season in September, when the above ground vegetation is already senescing or even has lost its leaves, there are still roots growing at this depth. So what the heck are these roots doing there actually at that late time in the year? Well, we had some other parts of both this and another experiment where we then really late in the year, in September or even in October, injected some 15N, so an isotopically labeled nitrogen, into the bottom of the active layer. So again, you know, late in the season at a depth where you would normally expect no roots to be. And then in the next spring, we looked at how much of this isotopically labeled nitrogen we could find back in the leaves of these species. And you see that there are several species, especially Eriophorum and Rubus. So that is cloudberry, and the first one is cotton grass, and they take up that isotopically labeled nitrogen from 0.5 meter down into the soil. So I started to think, like, okay, if they do this in September and October, when the above ground plant is sort of, by our perception, completely sort of senesced and ready for winter, what are they doing through the winter? And so this is basically how our adventure started. So yes, the winter is long. It is cold in some parts of the winter, and it is dark in some part of the winter. But actually, a lot of the landscape is covered by snow, and that doesn't only help us to get a sense of like it's actually not that dark, but it actually is also a really good thermal insulator. So on the right you see this graph where you see sort of the thick fat line in the bottom is the temperature that you can measure in the soil if you have about 1 meter of snow, while the bottom sort of zigzagging line is when you have only 10 centimeters of snow. So the more snow you have, the more stable the soil temperature gets, and the more likely, of course, it is that some microbial and plant processes can continue. And if we then think that about up to 90% of plant biomass is actually located below ground in a lot of Arctic ecosystems, and all microbes, also all soil microbes are, of course, also in the soil. So they are underneath that snow. You can imagine that actually maybe in the tundra things are happening in winter. And then combined with the fact that climate change, it is already happening faster in the Arctic than in most other places in the world, but it's happening even more rapidly in winter and spring and autumn. And these are parts of the year that actually most people don't do very much in the Arctic. So we wanted to change that, and we said, okay, let's explore some ecological activities in the Arctic winter and try to figure out what is going on there. Are these plants and microbes asleep or not? So I will show you some results, some preliminary results that we're working with from this big project that we're running. And so the first question is then, okay, how much does the cold season contribute to our annual CO2 emissions? And to do this, we went out into the field, and you need to get to your ecosystem. So quite often, yes, there is a lot of snow, so it's some hard work to get there. But then we had our postdoc Josephine Wals out there with her instruments to actually measure how much CO2 is coming out from these ecosystems and how much do they take up. And she did that every month throughout the winter, and then in the summer even more often. And so this is then the results that you would get if you, like most people, would be there only in the summer. In the top, in blue, you see the ecosystem respiration, so that's the CO2 produced by both plants and microbes. And in the bottom, you see the ecosystem productivity, so that's the CO2 that plants take up. And what you see is that in the middle of the summer, these fluxes are both largest, and then in the winter, we kind of assume that they're not happening. So is that true? Well, this is what you get if you also measure them in winter. They're not very large. They're definitely much smaller than they are in summer. But then the winter is so long that actually they still add up to a substantial amount. So we calculated that they are approximately 15% of your annual CO2 emissions. And this is then for two sides. On the left side, we have Abisko, which has a bit less snow, and on the right side, Vassajal, which is a location a bit closer to the Norwegian border, where we have more snow. And both sides, actually, we came to around 15%. Okay, so that was for the whole ecosystem, the respiration. And then we wanted to know specifically about plants, because if other peoples have been looking at fluxes in winter, what you usually read is, okay, this is sort of what the microbes are doing, and they don't think that plants are doing very much. So we wanted to see how much of this CO2 comes from the plants and how much comes from the microbes. And are these plants maybe also doing other things in winter, for example, taking up nutrients? So the picture that I will now show you is a little bit complex, so I will first explain the axis a little bit. So on the y-axis, we have the fraction of the plant respiration contributing to the total respiration. So if it's 0.5, which are the red lines, you would have 50% plants, 50% microbial respiration. And if it is lower than the 50%, we would have more microbial respiration than plant respiration. If it's higher than the 50%, we would have more plant contribution. And so this is, again, I sort of made gray what would normally not be measured in the winter. So if we only look at the growing season, we see that there is a little bit more than 50% of the respiration, so let's say 50 to 60% comes from the plants, and that means that there's 40 to 50% that comes from the microbes. And then this is what we found in winter, when we assumed that maybe plants would go down to, 0 or whatever, a very low percentage. We actually still measure 35 to 40% of the respiration coming from plants. So they are a bit less active, relatively speaking, than the microbes. And of course, both of them are less active than in the summer, which we saw on the previous graphs. But it's not that plants are just sort of dormant and asleep the whole winter. They are still active in these processes. So then let's look at the uptake of 15N. And this we did again by injecting isotopically labeled ammonium nitrate solution in the soil every month throughout the full year, and then coming back after a few weeks and taking a sample out and analyzing how much was taken up in the roots. So we looked at the whole shoot, the coarse roots and the fine roots of different plant groups. And here I've only selected the evergreen shrubs and the graminoids. And so if you would only do this in the summer season, you would get this picture. So you would see there's uptake. Some of it ends up in the whole shoot, but most of it stays in the root system. And you see that in the middle of the summer, sort of July, the uptake rates are highest. And so our expectation was that it would probably go down in winter. But instead, we found even higher uptake rates. And now I should say that this is, in a way, potential uptake rates, because it is what we inject and then what we see that they take up. And there are different assumptions that you have to make this fully quantitatively, which we are still working on. But you could say that this is sort of a potential uptake rate of the plants. So it's the potential of the plants to take up nitrogen, which seems to be even higher in winter than in summer. So, yes, that is sort of a little highlight of what we're working on now, and we got a big funding from the Wallenberg Foundation to continue with this and focus way more into both the plants and the microbial processes underlying this. So more to come. Thank you.
Thank you, Ellen, and congratulations to the successful founding that you have got from this. I think it's so fascinating to see more and more research are investigating dark conditions and seasonal variations and so on. It's
absolutely,
so
yeah. I saw it several times in presentations today coming back. So
we need winter data.
Yes.
Yeah. Yes, a lot of questions. Love and then Hanna.
Now?
Yes.
Okay, I'll repeat it. So there is this theory by American scientists who are trying to resurrect the woolly mammoth, and they're arguing that if they can release hundreds of thousands of mammoths into the Arctic, they will trample the snow, and this is going to reverse or at least mitigate climate change.
Exactly.
Since you are an expert on this, I hope, is that correct? If you have animals trampling the snow, will that actually lead to a reduced emission of carbon dioxide from the permafrost?
I mean, partly, and there are some data that support that. They have done some experiment. They have this Pleistocene park in Siberia, for example, and there have been some colleagues from Umeå University that have data from reindeer exclosures, for example, where you see that when you have reindeer, the snow is more trampled. You have lower temperatures in winter, so you have basically colder soils that could even end up in being permafrost. So in a way, that supports the theory that having lots of trampling on the snow could lead to the sort of reappearance of permafrost. It is the question whether that would be effective enough with the speed of climate change that we are producing. Also because the question is, okay, how high density of mammals do you need to have then? But yeah, also related to the data that Lena Ström this morning showed, with the fact that you have the disappearing of the moss layer, for example, with more trampling. So it could work partly, at least, I would think. Yes.
Now, Hanna.
Thank you for an interesting talk. I had a question. When you are doing your measurements, if I understood it correctly, you have to remove the snow.
Yes.
So this is a little bit related to the pressure, but do you think that removing the snow is affecting the activity of the microbes?
Yeah, this is a very good question, and that is one thing that is with many of our scientific things, that as soon as you start measuring it, you start disturbing the system. So ideally, if we have enough snow, we try to dig next to the plot and then come in from the side so that we still leave the snow structure intact. But unfortunately, quite often the snow is not deep enough for that, and we will have to basically take the snow away and then after our measurements put it back on to hopefully have at least part of that insulation restored. We work with different setups, so the carbon fluxes that I showed, they are from plots that are permanent on the same spot throughout the winter, so where we have to go back to the same spot. The nitrogen labeling we had a different spot every time, so the only thing is that we have to disturb it to inject the label and then come back several weeks later. But the measurements for the next month are done like a meter further away. Yeah.
Yeah, was it a question from Katarina? Or? Yeah, a question here.
You said that when you are measuring the nutrient uptake, you inject them with nutrients. Did you do that in the summer as well? So that
Yes, exactly.
Okay.
We give a very low, so the amount of nutrients that we add is really low, so it's way below fertilizing levels, but they are highly enriched with 15N, so that we can trace with the isotopes where it goes to. And every time we've been sort of injecting it and then coming back to that exact spot a couple of weeks later, and then the next month on a next spot. And so we've been doing that the whole year round.
Okay.
Yes.
Thanks.
Question down here as well.
Yes, thank you for your presentation. Yeah, I was wondering about the nitrogen injections. Did you inject the nitrogen in different depths, or?
No, not for this particular experiment. We had enough samples to deal with already.
And which was the depth that
we tried to inject it sort of over the range of 0 to about 7 centimeters.
All right, so in the topsoil. Yeah.
Yeah.
Yeah, thank you very much.
Okay, thank you, Ellen.